Alright class, so we last left off with the cardiac cycle. Just want to finish this lecture and then move on to the next one covering the nervous system. Alright, so this might be a little bit of a rehashing of the previous lecture slides, but let's kind of move on forward here. Okay, so we mentioned last time, I keep mentioning the word cardiac cycle. So let me go over how this looks like. So on the left you have the heart and all the chambers, and the red represents how flow is going through the heart. These little green lines are the valves, designating if they're open or closed. And so let's go over, there's generally two phases of the cardiac cycle. Remember that there's systole and diastole. Systole is the active contraction of the ventricle, and diastole is the filling of the ventricles. You can break them down into 4 phases. So what we'll do here is we'll designate this first phase here as ventricular filling or diastole, but what I'll call it is the end of diastole. So in the middle here you have a plot of the pressures in the ventricular chambers, the atrial chambers, and also the great artery that's connecting to that ventricle. So these plots look the same on the left and right. The main difference is the values of the pressure are much lower on the right than on the left. So for here we're going to focus on the arterial side, or I should say the systemic side here. On the bottom here you have a plot of the volume of the ventricles in time, and the x-axis is time. So this is occurring within 800 milliseconds for your cardiac cycle. So let's go over it here quickly. One more thing. On the right you have the pressure volume curves for the ventricles. So what that means is if I plotted the red line versus the black line, which is the ventricular pressure, and within this you do have time, but you can't see it, so it's not explicitly shown here. But let's say you start at A. In time you'll go around this loop here, and that would designate back to one cardiac cycle. So you're kind of going counterclockwise in this loop in time, and that would be one cardiac cycle. You keep going around that loop. Alright, so let's start off here. So during the end of diastole you have filling. So what that means in terms of what's going on with your heart is, on the left side at least, that you have the left ventricle is filling up. What that means is that the pressure in the left atrium is bigger than in the left ventricle. That causes flow to go from left atrium to left ventricle because of that pressure gradient. But at the same time you also have that the aortic pressure is bigger than the left ventricular pressure. So that means that you don't have this valve open because there's no pressure gradient to push it open. So what that looks like on the pictures on the in the middle here is that the dotted line on top here is the aortic pressure. That is bigger than the left ventricular pressure, which is shown in the red. The dotted line on the bottom is the atrial pressure, and you can tell that it's barely higher in value than the red line. So that means that the pressure gradient is pushing the ventricle to fill up. In terms of volume, you see that there is an increase in volume from approximately 100 to 120 milliliters of volume. So where on this plot is that? So let's say that you're mapping out the volume is at about 100. That's approximately right here. Volume's increasing a little bit, but you don't have much of an increase in pressure in the ventricle. It's kind of seen here by this really small hump, but not really too high. So that's right here. Okay, so going from this point here, somewhere between B and C, and more towards C, all the way to C. Alright, so once that happens, you hit the end of diastole. That's the end of the filling phase. At this point, you have the ventricles that will start generating pressure to eject, and that is the next part of the cycle here. So this is part of systole. So it's actually the early part of systole, and it's called isovolumetric contraction. What that means is that iso means that there is no change in volume, but you do have an increase in pressure. So here, what that means is that all of the valves are shut. So the left atrial pressure is now smaller than the left ventricular pressure, but because the valve does not allow flow to go backwards, there is no flow to the left atrium. But you still have left ventricular pressure is below aortic pressure, so you still have the valve closed in this case. But now the muscles, the myocardium in your ventricles are contracting and causing a buildup of pressure. On the plot here, you see that the red line finally goes over the dotted line on the bottom. So that just tells you that now ventricular pressure is exceeding atrial pressure, which causes this valve to close. But you haven't hit it, it hasn't gone above aortic pressure just yet, so there's no ejection. What you see on the PV loop, the pressure volume loop, is that you have an increase in pressure, but no change in the volume of the ventricle because there's nothing leaving the ventricle. So that you'll see that from say 10 millimeters of mercury all the way up to on the order of 80 millimeters of mercury. So that's that isovolumetric contraction. In terms of volume, see, nothing changes, right? It's still at about 120 milliliters. Alright, so the next phase is basically the rest of systole. And what you'll see here is this is finally when the left ventricle pressure exceeds the aortic pressure. But it's still above the atrial pressure. So you see the red line finally goes above the aortic pressure, so that causes flow to come out of the left ventricle and go into the aorta. And it's again still higher than the left atrial pressure, which keeps the valve here closed. That also means that the volume is going to be changing in the left ventricle. So as it's ejecting fluid out, you have a decrease in the volume of the ventricle. It doesn't go to zero, but it's almost there. And notice how you have this LV-EDV and LV-ESV. This is left ventricular and diastolic volume. This is left ventricular and systolic volume. This is the volume in the ventricle at the end of systole, so once you've fully contracted. Again, mapping the red line with this black line at the bottom here, you'll find that you're going from D to F. So you do have an increase in pressure, obviously, and then you're also now ejecting the volume back from 120 to 50 milliliters in this case. So that's where you end up down here. And the point F here is the end systolic volume. That's right here. And the end diastolic volume is sitting at C. That's the one up here. Okay, so that's ejection. And then what you'd have next is isovolumetric relaxation. So that means that now your muscles are relaxing, so you're gonna want to eventually fill. This is the early portion of diastole, and essentially what you have is the pressure is decreasing in the ventricle. It decreases below the aortic pressure, but because this valve does not allow backflow from the aorta to the ventricle, this valve is closed, but you're still above the atrial pressure, which again, this valve here does not allow flow to go from ventricle to atrium, so all valves are closed, which cause a decrease in pressure, but no change in volume. And that's seen here. This line, no change in volume. And then so finally, you have, diastole is the rest of this, right? It's this whole part here, where you start at A, go to somewhere between B and C, and then you start all over again. So this whole loop entirely tells you how pressure and volume is changing in time, and just make sure you know the different pressure gradients for the different parts of the cycle. Okay, this plot does have some validity in terms of what it tells you about the heart and how it's working. It turns out that the pressure volume loop does give you the amount of work that the heart does in one beat. So if you take the area underneath this curve here, within this curve, this is the amount of work the heart does, and the metric is joules. So on the right side here you see that in equation form. So the amount of work is equal to PdV if you integrate with respect to volume, because pressure is a function of volume. you're going to get basically that work is equal to delta P delta V. For some numbers here, I just crunched some numbers, assuming this was a box. Obviously it's not. You can guess that, the amount of pressure that you're generating on average is about 105 millimeters of mercury. Converting that to pascals, the amount of volume that is changing is 70 milliliters, approximately, from 50 to 120. And then you convert that to meters cubed. You're talking on the order of 1 joule of work or energy that is created by the heart. Now, if you want to get wattage, you just multiply that amount of work times the heart rate. And so for, let's say you have 60 beats a minute, so 1 beat per second, that would give you about 1 watt of power. So your heart is outputting 1 watt of power approximately per cycle, which is quite a bit of work or energy you have to output. The other metric you can get out of the pressure volume is the stroke work. Remember that that's the difference between the end systolic volume and the end diastolic volume. Again, this is just to put some numbers behind what kind of work your heart does. And obviously it changes depending on the blood vessels attached to it, the amount of pressure generated, and volume. So what I kind of want you to take away from this is kind of the numbers and how we calculate it all, but also that the shape of this thing does depend entirely on the amount of pressure the ventricle can generate, but also the blood vessel attached to that specific ventricle. So we'll get to that a little bit later, I think in the next slide actually. Alright, so here's an example of showing you the different pressure volume loops for a diseased heart versus a normal heart. So here in the yellow you see that that's the loop for a normal heart, and then the pink is what you see as a diseased heart, which has lost its ability to contract, basically. So ionotropy means its ability to contract and create a force. Notice that the amount of peak pressure goes down. So like here you're on the order of 120, here you're on the order of 110. Notice there's a shift in the volumes now, right? The end diastolic volume is now 100, and the end systolic volume is here. I'm sorry, other way around. End diastolic volume is about 150, and end systolic volume is on the order of 100. So you have a lot of extra volume that's within your heart. What that means is that the extra volume has to be accommodated for, and so that results in a larger volume in your ventricle, which over time will lead your ventricle to expand. So this volume shifting is what you're seeing as basically your heart has to basically increase in size to accommodate for the volume that you're accounting for. Notice also the difference, right? The control loop has your stroke volume. It's much larger for the control loop than it is for the red loop. That's because you have less volume that's being injected out per cycle, and that has to do a lot with the contractility of the heart, but also what's attached to it, so like the blood vessel. So to get around this, we use ventricular assist devices. So the way it works is you want to get this red loop to be shifted back to the left, and to do that you're going to have to unload this volume. And so to do that you would attach a pump at the left ventricle to basically drain out the volume that's extra here, and then pump it to your aorta, and hopefully that will help you with this PV loop. So obviously if you're working in this kind of research space, you have to know a little bit, or actually a lot of it, about the pressure volume loops and what they correspond to in terms of the physiology, but also be able to predict how the device that you're designing and using is going to affect that volume loop, and if it helps the ventricle, or it doesn't help the ventricle. Here's some questions to kind of consider if you're working in this kind of research space. What kind of power outputs are needed from a device like this? What kind of pressure differentials must the device work against? You already know that like this pump has to see a pressure difference of on the order of 0 to 120 millimeters of mercury. So this device has to work against those pressures, and it also has to be able to unload, a couple of milliliters of volume from a space. Um, this is why I put numbers to those plots, because if you're designing any kind of system that interfaces with biology or physiology, you have to know what ranges you're working with. The other thing is, if a regular heart outputs 1 wattage of power, what does your pump have to output in terms of mechanical power? And that correlates to the kind of batteries you need depending on the efficiency of your whole entire system. So it's all connected, right? I mean, knowing the physiological parameters you're working with affects the design of your device, but it also affects the kind of power that you're going to have to generate and also supply to the electrical components of it. So this is just giving you a rough idea of what you have to expect. Okay, this is a part of a paper by Thiel where they study 3 different types of blood pumps, and seeing how they affect the pressure volume loops for the patient. So the green lines are the pressure volume loops corresponding to the disease state of the person. The blue lines are showing you how exactly the device, once it's implemented in the patient, affects the pressure volume loop. So there are these devices called intra-aortic balloon pumps, which are basically just what they look like, balloons that are placed in the aorta, and they basically are pulsed and kind of out of phase with the heart. So whenever the heart's contracting, this thing is deflated. When this is filling up, this thing inflates. And what that helps to do is it helps to get rid of the volume that's within this area where the balloon is. So this is called intra-aortic balloon pumping. You also have intravascular blood pumps. So these are little turbine pumps that suck blood from the ventricle and suck them to the aorta. And this is going on during the entire cycle. You also have percutaneous blood pumps, which are pumps that you can access the veins and arteries and then get that volume and put them through the pump. And so let me just show you how some of these devices affect the PV loop. So in the case of the intra-aortic blood pump, you see that the disease state, you have pretty large end systolic and end diastolic volumes. When you have this device, it unloads this volume in the aorta, which naturally then decreases the end systolic and end diastolic volume, because there's an ejection of volume here, so this causes this to drain out more easily. So that's why you have a leftward shift, and that's all it changes basically. You're just shifting this to the left. So in theory, if you had a bigger balloon here, like say all more volume, you'd be able to then move this blue line even further to the left. We'll get to these lines later when we talk about modeling, but they're not important right now. Okay, in terms of intravascular pumps, it's a little bit different, right? It actually changes not only the end diastolic and end systolic volumes, but it also changes the shape of this thing. So in this case, you're generating more pressure, seen up here, but you're also making the stroke volume smaller. So here we didn't change the stroke volume much, because the difference between this line and this line and this line and this line are not very different. In this case, you are changing it a lot, right? So this does unload the ventricle, and it does so by decreasing the amount of stroke volume that the ventricle has about. That's because you're sucking volume directly from the ventricle. So when the ventricle does contract, it's not contracting a lot of volume because it's being sucked out by the pump. But you do see that so that does change the diastolic volume quite a bit. Not the systolic volume so much, but the stroke volume is changed quite a bit. But the pressure is not changed. So, if you have hypertension or high blood pressure, this is not gonna change it very much. So just that's something you have to consider. In the percutaneous pump case, you don't have direct drainage from the ventricles, so you're not gonna see this drastic change here because you're sucking direct from the ventricle. You will have a change in stroke volume. Your end systolic volume will actually increase. So you'll have some leftover blood in the ventricle, but your end diastolic volume will decrease. In total, your stroke volume goes down. So that's the area here. And your blood pressure actually goes up, and that's because this is providing an extra resistance for the left ventricle. So this actually does increase the pressure. So again, not good for hypertension. So again, things to consider when you are using different devices. They affect the heart a different way. Okay, measuring hemodynamics. So this is a short couple of slides on how to actually measure these things. Here's a quick history of blood pressure. So in 1777, this is what I was telling you before when they first measured blood pressure. So Stephen Hale measured blood pressure from a horse, and here he used a tube with mercury inside of it, and you have to access the vein or the artery, and he was able to do that by putting the tube in the artery, and he saw these weird pulsations. So you'd have an increase in the height of the blood and decrease in the height, and that's what he did. From then on out, obviously we've had much better technology to do that. You don't have to poke a tube in your artery to measure it. The mercury manometer is invented, so that's the kind of the U tube, not the U tube as in like the videos. Like you have the U-shaped tube that has mercury inside of it that allows you to measure the height. And Mr. Carl Ludwig invents an apparatus that allows you to actually map out a plot of the pressure and time to do so. So there's a quick history of that. But what do we have now? So the most common way is to indirectly measure the blood pressure. So don't actually have a tube interfacing with the blood per se. We have this cuff and manometer-like setup here. So what you need is what's called a stethoscope. That's so you can hear some of the sounds coming from the blood vessel. You need a mercury manometer and an inflatable cuff. And so the way it works is like this. The doctor will, generally put a stethoscope on your arm. He will inflate the cuff. They'll inflate it so much that you feel like your arm's going to fall off. But in fact, they only inflate it such that there's no sound coming from your arm. Once that happens, that means that the vessel has totally collapsed, so there's no blood flow through that vessel. Now remember, you have lots of vessels, so it's not like you're going to die in this case. And once he hears that, he's past the systolic pressure. So here, the doctor, he or she will then let go of the cuff pressure, and as the cuff pressure is released, it opens up the blood vessel, and with that, you'll start hearing some sounds. And they're called Korotkov sounds. And so the first one you will hear is your systolic pressure. That's like the swishing sound when blood finally starts to go forward. That's systole. That's the highest pressure your ventricle will generate to move blood. And then you'll keep hearing some sounds, some lub-dub sounds, and once that sound disappears, then he or she will mark that sound, and that'll be your diastolic pressure. So that's when fluid moves freely and you can't really hear much with the stethoscope. So those are the two numbers that they're basically hearing. They're hearing the first sound with the initial opening of the artery, which is about 120 millimeters of mercury, and then the last one is the last sound you hear, which is at diastole. Obviously, you can automate this. You don't need a person listening for sounds. You can actually have some piezoelectric measuring, some microphone measuring that. You can have an automated system that inflates this above a certain pressure such that the microphone doesn't hear anything. So all sorts of things that have already been done to make this more accurate. All right, the other method to measure pressure is invasively. So this is having a direct connection to the arteries or the veins. So what you'll need here is an arterial line. So it's basically a needle and some tubing that can have blood inside of it without coagulating or being damaged. You need some saline, which is basically like ionic fluid, a flushing system to flush out blood so it doesn't clog up the tubes. You need a pressure transducer to actually measure pressure. And so the way it usually works is if you're on, the bedside in a supine position, so like laying on your back, you have a tube connected to a pressure bag as your reference pressure, and the transducer will then, this is to flush out any blood, and then blood will interface here with saline. saline and blood have around the same viscosity and density. So you can measure the pressure you measure down here will be measured up here. And that transducer will then measure the pressure fluctuation in the line on a monitor. The transducer is pretty important. Usually you use the strain gauge to measure pressure, and the way it works is like this. There's two types. There's unbonded and bonded. So what you'll have is you'll have a diaphragm that's interfacing with the fluid, in this case saline. When pressure is applied, it'll move this diaphragm, which will move this like little apparatus here that's connected to wires. And these wires are strain sensitive, so the resistance of these wires that are connecting this movable object to the housing of the transducer will flex, and the amount of strain will change the resistance of the wires. So you can then, basically measure the resistance changes for different applied pressures, and that gives you something to calibrate, and you can then convert, resistance changes to actual pressure readings. The most common type you'll see are called bonded blood pressure transducers. The way these work is on the diaphragm. Instead of having this weird mechanism here, you have this foil pattern on the backside of the diaphragm, and the foil pattern will have like this snake-looking geometry. That's just to increase the length of. This is basically a resistor, and you have some wires connected there. And this is basically a variable resistor, and the variability in resistance depends on the strain, which depends on the pressure being applied on the diaphragm. So that'll be placed, and then when this diaphragm moves, it causes a change in resistance, which then gets calculated to a change in pressure, which you'll see on a screen. One important metric for these sensors are the gauge factor. That's just basically the amount of resistance change over the original resistance over the length change over the original length. So this gauge factor is an important metric when choosing different strain gauges for different applications. But this is how you would basically measure pressure. This gives you the highest fidelity pressure signal because you're measuring directly within a blood vessel. I'm not going to get into this, but this is a way to convert that resistance change into an actual voltage reading. And let's go over to the bonded strain gauge here for a second. So the way to do this is you're going to have to then get one of the strain gauges in your pressure sensor, and you're going to have a voltage source and a bridge of resistors to help measure that resistance change. It turns out that you're going to pick resistor values for the strain gauges and these resistors that are all the same. That's going to produce a voltage in the middle here that is 0 volts if these are all the same value. That means that the bridge is balanced. When you change the resistance of this, that'll change the voltage measured across these two wires here. Now the reason why you have two of these is because these are also temperature sensitive. So any temperature changes will cause a change in resistance. So in order to get rid of that, you're going to have two strain gauges. One of them is unstressed, so not on the actual diaphragm. It's just feeling the same temperature changes as this one here. So any temperature changes will be canceled out by having two sensors that are at the same temperature. We'll get more into this in 503, but I just wanted to introduce it to you because, it's for those electrical engineers that are bored of physiology. This gives you some context of how to actually get to hardware that helps you measure like physiological parameters. Okay, there's a couple of ways to measure cardiac output. I'm only going to go over, I believe, one of them, which is not as fun as like Doppler echo and all that stuff. So we'll go over that in 503. But one of the oldest ways of measuring cardiac output is called the indicator dilution method. The way it works is like this: the doctor will inject dye in the venous circulation. That dye then gets distributed out to the rest of your circulation, and just based off of a conservation of mass, assuming that no dye was consumed by like any of your organs, you can calculate the amount of flow from the area of the dye concentration at a specific point. So, like for instance, like say they'll inject dye at a certain point in circulation. Then they will also extract some blood and then test it for like the amount of dye that's in that portion of blood, and there then will be able to get a curve of the dye concentration versus time. So a T1 is where they inject the dye, T2 is where, at some point downstream where they measure it, and the concentration will go up initially, but then the dye will distribute to different parts of the body and then basically get excreted by the body, and you'll get this curve where you have an increase and then a decrease in dye. The area under the curve gives you the instantaneous dye concentration, that's right here, and that over the amount of dye you put in, the amount of dye you put in over the area under this curve will give you the flow rate. So obviously for a higher flow rate, this will be a much smaller curve because it reaches it much quicker, and then like T2 will be over here versus over here, so that area will be smaller, which will give you an increase in the flow. So that's one way. The most common way nowadays is this indicator dilution method is really old. They use a thermal dilution. So instead of actually injecting dye, they'll induce a temperature change in the blood and then measure the temperature change somewhere downstream. You'll get a similar plot, but instead of dye concentration, you'll measure temperature, and you'll have a similar curve, and the area under that curve will then be used to measure that. So that's called thermal dilution method. That's the most common way. This method doesn't give you nice waveforms. It just gives you an overall metric of like how much liters per minute is output by the heart. If you want an actual like wave in time, you're gonna have to use other methods to measure flow. And in fact, to this date, there aren't many commonly used methods to measure flow rate or cardiac output that can give you like an instantaneous measurement. You're gonna have to use what's called echo Doppler, or you're gonna have to use some other method like a flow meter, which can be pretty dangerous in terms of interfacing with the blood. Okay, so let me move on to at least talking about some of the neurons in your body and the nervous system. At least introduce it. So let's go to week 4 lecture. So this is the last set of lecture slides that go focus directly on physiology and anatomy. We are going to have some lecture slides that go over like bioelectric phenomena, but at the end of the day, we're going to just stick to this to be the anatomy and physiology we cover. So let's go over a little bit of it. So one of the most important organ systems, which you no doubt know about, is the nervous system. I mentioned it before. The nervous system is responsible for integrating all of the functions in the heart. So this goes into integrating the circulatory system with the respiratory system, with the digestive system, etc. All of that's integrated within this kind of control center. You have a network of trillions of nerve cells that are linked together to basically allow for you to respond to different stimuli that you are affected by. So for example, we can break down the nervous system anatomically. The central nervous system is all nervous tissue and all neurons which are enclosed by bone. So that includes like your brain and your spine. Your spine has lots of nerves in it and nerve fibers which are enclosed by the spinal cord. Everything else is the peripheral nervous system, so that's all nervous tissue that's not enclosed by bone. That includes like sensory neurons for touch, for taste, for seeing things, and other neurons that connect to the central nervous system. So this is how they are anatomically broken down. Now I'm going to go over some ways to break it down physiologically, which is the next here. So you can actually break down the nervous system into two different parts, but based off of function. Remember that anatomy and function are not the same thing. So we break down the nervous system in terms of somatic and autonomic nervous system. So somatic means voluntary movements. So these are nerve fibers that control muscle voluntarily. So like when you actively lift your arms, or as I actively talk into this microphone, I am doing so voluntarily. So there's something going on in my brain that is sending signals through the nerves that connect from my CNS to my sensory receptors and to my muscles, which allow for controlling some, movement in my mouth, for instance. So you'll see this a lot in this next couple of slides. This is a cross section of your spinal cord. You have some of these um, connections here. These bundle of neurons connected at specific parts of the spinal cord, and you'll see like this light white and this kind of gray matter. We'll get to that a little bit later. But this is a cross section of your spinal cord. You have lots of these discs. But this is just showing you you have some coordinated movement that originates at your brain up here down to your spinal cord, and then goes to an actual neuron, which then affects some muscle. And there's various ways that it can control it, but we'll get to that later. The autonomic nervous system regulates involuntary processes. So this is like your breathing, your digestion, anything having to do with your everyday kind of processes that we kind of take for granted. We don't actively breathe. We can actively breathe. So breathing is one example of where we do have some nerves that are somatic and some that are autonomic. But autonomic, that's where we breathe on an everyday basis without thinking about it. And on a similar note, you have some processes that's in your brain that are controlling that go through your spine, and then there's some connectors here that control your breathing and your heartbeat, etc. Just an example of how that kind of works. Okay, I'm gonna end on this note. Let me actually just skip it here. I'll end on this note here. Yeah, I'll go before I get to central nervous. So I'll end on that. So, what are neurons? I keep using neurons and nerves. Let's define them exactly. So neurons are the functional unit, the functional cell of a nervous system. You're going to see this picture a lot in the next couple of slides, but neurons can have all sorts of forms, can have all sorts of dendrites and nuclei, so we'll get to that later. But this is basically the fundamental cell for the nervous system. Nerves are just bundles of neurons. So if you have a bunch of these neuronal cells that are connected together, and they all bundle to like some fibers that will send signals from cell to cell. Okay, so a couple of things that you might have seen like in high school physiology. Dendrites. Dendrites are long processes that extend from the neuron cell body. So this is the neuron cell body. This is the nucleus. These here are called dendrites. These allow the cell to communicate with various other cells and various other types of cells as well. They increase the surface area of the neuron, and again, they help communicate with multiple cells. The region between the dendrites and other cell processes are called synapses. So they're right here. So let's say you have this cell extending its dendrite to another dendrite. The area between here is called the synapse. That's a region where axon terminals, which is the other end of a cell, meet a target cell, and the narrow space between there, on the order of like nanometers, is called the synaptic cleft. This is where like things like dopamine and other neurotransmitters are released, and you have communication. You have chemical communication between cells. Notice on the cell you have this other long process, kind of like a long wire, that's called an axon. The axon can have this thing called myelin. It's basically fatty molecules that are surrounding the axon. When a signal is generated from the nucleus, it propagates along the axon, and this myelin, called myelin sheath, can act like an insulator and allows for that signal to go through without coming out of the sides of this axon um, length here. So that's important. Some neurons will have this myelin, some won't. So we'll get to that when we start modeling different parts of the body. Let's see what else here. So I mentioned the axon. That's the long thing here, myelin sheaths. I think that's basically it. So one take-home message I wanted you to take from this is that neurons can communicate electrically, and we'll get to how you have electrical signals propagating along this length. But they also can communicate chemically across these synaptic clefts. So just give you an idea of how these things function. This is just showing you that there are different types of neurons, depending on if you're in the central nervous system, the peripheral nervous system. You can have, like the ones I showed you, where the cell body's in the center, and then you have some multiple axons here, and then the dendrites. You can have, no myelin sheath. You can have just cell body and just dendrites. You can have a tree here, like of dendrites. It really depends on the type of neurons you're dealing with and where in the body you are. But take-home message here is that there are just all sorts of neurons, depending on where you are. Let me just show you how, like a functional diagram of how your nervous system works, and then we'll end it here. So, you have your central nervous system, which is your brain and your spinal cord. You have some peripheral nervous system, which deals with your sensories and all that stuff. So let's talk about what happens when, how your brain splits up tasks. So let's say that you got your hand and you touch the hot pan. That is a signal. So you're gonna have your receptors on your hand. They're gonna feel heat, and there are some ion channels in your fingers that will open up and cause an inflow of ionic current, which will then be an electrical impulse that gets sent directly to your brain. Now here's what what's not really shown here. This signal here actually goes to your spinal cord first. And then that gets thrown up to the central nervous system. So you have some thermoreceptors which feel that. What's kind of misleading here is that it actually goes to the spinal cord. That causes you to take your hand away from the pan, which is something that you don't have to think about. It just happens. That job is for the spinal cord. So that's kind of the relay station. If there's any reactions that you have, that gets processed by the spinal cord, and then once it hits your central nervous system, then you realize that you burned your hand. Especially two pathways that are not really controlled here, that are not being shown. I think I'll have another picture of this when we talk about the spinal cord. But what this also does is it doesn't just change. So I guess the point of this slide is to show you that it changes your somatic and your autonomic response. So I guess once you realize your central, that you're burning your hand, you control your muscles to remove the hand, which is the primary responsibility of the spinal cord. But then also the brain changes things that you're not really aware of, like your blood pressure changes. Maybe you increase your blood pressure, your breathing changes, the rate at which it's a changing also will be affected. So all of that is processed by the brain, and there are things that you can control and things that you can't that just happen. So I'm gonna end on this topic, but one last thing is this is a good place to mention. Even stress can lead you to have these autonomic changes in your central nervous system. So when you have anxiety, or you're prepping for a test and you're getting worried about it, that can also increase blood pressure, because that is being seen as like, oh, something is stressing me out, whether it be at a over a chronic amount of time or acute amount of time. Either way, it can change the blood pressure and the breathing patterns. So that's why it's important to basically be aware of that kind of stuff for your brain. I think that's all I wanted to mention here. But again, we'll see something like this similar, but we'll break it down into what the spinal cord does versus what the brain does later on. Yeah, so I mean, from here on out, we're just gonna go over some of the anatomical aspects of the brain and the central nervous system, but we're not gonna get into too much detail. We'll finish this off on next Monday, and we'll also get into the cochlea and the eye, and then hopefully we'll get into compartmental modeling at the very least next week. Okay, be on the lookout for the LaTeX video coming up soon. Forgive me for the long video recording.